So this is our second organelle video, and we're going to start off with a fun organelle, which are ribosomes. Ribosomes, as we're going through which cell types these are found in all eu and prokaryotic cells, so both bacteria and our eukaryotic cell types. Ribosomes look like small circular structures, and they are found in one of two places. They're either free-floating in the cytoplasm, or they're embedded in our endoplasmic reticulum, which is another organelle type. The ribosomes that are in the cytoplasm are typically making things for use inside the cell, proteins that are going to be used inside the cell, and ribosomes that are embedded in the endoplasmic reticulum, or ER, are typically making things for export, things that are going to leave the cell. This is where all of your proteins are made. And what they're doing is they're going to translate RNA, which is made from DNA, into amino acids. So pictured here is a ribosome. Here's a strand of what's called mRNA or messenger RNA. Okay, the ribosome reads the code here, the little bases here, and it makes the protein. This is the protein being made, coiling away off to your left. We're going to get really detailed in this process in Unit 7 or 8 later in the year, but for now we're just going to be worried about what the ribosome's job is, which is making proteins. So this slide is showing you right here, here's a piece of mRNA coiled right here, and all of these dots structures that you see here are individual ribosomes, okay? So you're seeing individual ribosomes attached to a single direction, set of directions to make a protein, and all these little structures you see coming off are proteins that are being formed. And remember, these proteins could be anything from enzymes, things involved in chemical reactions, to hormones, okay, messengers between cells, to structural proteins, things that would make up hair, nails, muscle fiber, cartilage in your skeletal system, etc. And these are all being made by these ribosomes. Ribosomes don't do a specific job, so a single ribosome could make many different protein types. Pictured over here then is an electron microscope picture of ribosomes embedded in the endoplasmic reticulum. So these individual specks that you see here are ribosomes that are embedded in the membrane layers of what we call the ER, the endoplasmic reticulum. What the ER is it's a series of membrane-bound vesicles, okay, or really tiny vacuoles, right, that form channels inside the cytoplasm. So if you look, it's like a maze. All these pieces are interconnected. It's like a big giant tubular network, okay? And what is happening in the endoplasmic reticulum is varied depending on the type. We have two types. We have, first of all, smooth endoplasmic reticulum. That's pictured out here at the edge. And this is ER that doesn't have any ribosomes embedded in it. The function of the smooth ER is to transport proteins and membrane lipids out of the cell. So you've got your nucleus, usually you'll have your rough and smooth ER, and the smooth ER is sending things out towards the membrane. We'll see that in a picture here in 1 second. The rough ER then is the part of the ER that has all these specks, these ribosomes embedded in it, and its job is to actually make the membrane proteins and the hormones and things for export from the cell. And so what happens is the rough ER makes proteins inside this membrane space. Those proteins are then transported over to the smooth ER, and the smooth ER then sends those items out for transport from the cell. Okay, so what kinds of things are made here? And once again, that's membrane proteins, secretory proteins, things like hormones, proteins that are going to be outside the cell, think like platelets in your blood, and membrane lipids are all made in this rough and smooth ER network. Our next organelle up then is the Golgi apparatus. Now the Golgi apparatus is this really neat structure. It's really the intermediate step between the ER, seen here on the left-hand side, and the outside of the cell. And so the Golgi is, it's a series of membranes. Again, I like to say they look like a couple of pancakes stacked together. And their task or their function is to process, sort, and modify proteins and other materials, things like enzymes, as they're going to be traveling out of the cell. And so the Golgi is tasked with taking the substances made in the ER, making any modifications that need to happen inside their intermembrane space here, and then passing those substances along so they can be merged with the cell membrane and released through exocytosis, EXOCYTOSIS. These sacs are oftentimes filled with enzymes. We call these intermediate, these little vesicles, we actually call them transport vesicles. And so they're moving things out of the cell. A lot of stuff actually needs to stop here from the ER, needs to stop in the Golgi so that it can get modified or changed in some way, the structure, so it'll work once it leaves the cell. This slide then just takes in the whole process of protein formation in the rough ER, transferring those substances to the smooth ER where they bud off, they travel to the Golgi, they're altered in the Golgi or edited, and then they're passed along for transport outside the cell into your body fluids, bloodstream, external environment, etc. Up next then are lysosomes. Lysosomes are this really neat organelle that's tasked with basically breaking things down. Remember, lys means breakdown. And so lysosomes are pretty much just found in animal cells. They contain digestive enzymes that are capable of breaking down almost any organic molecule. So food particles, organelles, pieces of virus or bacteria, all right, all kinds of different things. The lysosomes are actually made from the Golgi, so they're vesicles that have budded off from the Golgi filled with these digestive enzymes. They can actually kill the cell, so if you rupture a lysosome and release the enzymes out of it, they can kill the cell. That sometimes happens on purpose. It's called apoptosis or programmed cell death. But just really neat organelles responsible for breaking things down, recycling. Then we're going to look at the cytoskeleton. So this is the pieces of the cell that make up the structure and support. So if we look at a membrane here up top and then another plasma membrane down below, we've got all these components that are little bridges and scaffolds and things that are working across the cell, as well as these larger structures that serve as highways through the cell. And so what we have there, microfilaments, these smaller vertical things, these are microfilaments here, and then the big sort of tube structures, these are microtubules, right? Microfilaments are involved in movement, support of the cell, and then we've got microtubules, these bluish ones here, that are also for support, but also are what help to create that cytoplasmic streaming. So you can move organelles down microtubules with this really neat protein called kinesin, which I'll show you in class. An often overlooked part of the cytoskeleton are what's called the centrioles. Centrioles are basically anchor points made out of microtubules, and they serve to anchor and pull the DNA around during division. You wouldn't normally see these in a cell if it wasn't dividing, but they're responsible for coordinating the movement of the chromosomes or the genetic material during division. That's the centrioles. We also have microtubules that can extend out from the surface of the cell. Okay, and so we can have really one of two types. We can have what are called cilia. Cilia are shorter hairs present in usually very large numbers. You saw cilia when you looked at the paramecium slides. Or we could have flagella, which are longer and less numerous. You could have 2 or 3 flagella or more on a cell, but there's not as many from cilia. Usually only on like one area of the cell would a flagella be extending. But both of these are used for movement. Flagella will basically whip around in sort of like a circular torpedo or propeller type movement, and the cilia will beat back and forth, almost like oars on a ship. So this slide just highlights some of the different structures. These are cilia, electron microscope picture of cilia. This is cilia on a paramecium, and they can be used in the mouth parts too. So in the mouth parts of the paramecium, there's some cilia that push things into the feeding opening. And then here's some different bacterial cells with flagella. So here's a Spirulli bacteria with a flagella off each side. Here's a Bacillus bacteria with a single flagella. Here's a Bacillus bacteria that has 4 flagella, so a lot more in that case. But so this is flagella up here and cilia down here. Cilia short, flagella long. Up next are chloroplasts. Chloroplasts are really amazing organelles. They're found in eukaryotic cell types only, and specifically found in plant and protist cells, some protist cells, so plants and protists. Chloroplasts are amazing because they serve as the basis for almost every food chain here on Earth, and they basically use energy from sunlight, actual photons from sunlight, to make food molecules. We're talking about sugars. Okay, so they turn the sun's energy into sugars. That process is called photosynthesis, photo meaning light, and synthesis meaning make. And so that's a set of reactions that take sunlight and make food and oxygen from water and carbon dioxide. Where that's happening, or what chloroplasts are using, are a special pigment that we call chlorophyll. This is a green pigment that's responsible for trapping some of that energy. We're going to get really detailed with the structures of the chloroplast in a later unit, so we're not going to go too far with those right now. The interesting thing about chloroplasts, just like mitochondria, guess what, guys? They have 2 membranes, they have their own genetic material, their own enzymes, and so we think, just like mitochondria, that chloroplasts originated as an independent cell. Remember, this is the theory of endosymbiosis. Where chloroplasts also were engulfed by an early eukaryotic cell type. They were integrated in. Think about the benefits of this. What used to be a bacteria was now integrated into these cells. When exposed to sunlight, it made sugar for those cells. So think about the benefit to the cells of integrating the chloroplast as an organelle. This likely happened by mistake, but the cells that had chloroplast present were at a great advantage over other cells. This slide is just showing you really some of those substructures in the chloroplast, and it's also showing it's not just artist renderings that show these things, but this is an actual electron microscope picture of a cross-section of a chloroplast where we see these same structures that the artists are drawing. We're going to finish up talking about the cell walls. Cell walls are found in prokaryotes, remember bacteria, and eukaryotes. Which types of eukaryotes? Well, it's protists, some protists, plants, and fungi. Now, the chemical composition of those cell walls is different depending on what type of organism you're looking at. This slide is going to focus on plant cells. And the cell wall is this outer structure that gives the plant cell a lot of support, okay? It's a non-living layer. In plants, it's made of a starch called cellulose. We'll talk about fungi. It's made out of something called chitin, okay? And prokaryotes, it's oftentimes made out of something called peptidoglycan. We'll get to those later. But cellulose is actually a starch, okay? So it's a complex sugar, right? A carbohydrate. And it's found in plants in that form. And like I said, chitin and fungi. This is a very strong structure. This is basically what wood is, right? Tree trunks made out of cellulose. And so it's useful for a lot of building materials for the plant and for humans.